ce qu'ils vont, une mise chargée de recherche.
Okay, thank you very much for the introduction. And first of all, thank you very much to the organizer. I'm very happy to be here, and I will be very pleased to present you some recent research about chemotaxis modeling and analysis of chemotaxis equations. So, in the spirit of Michael Shelley's talk this morning, I will discuss chemotaxis for bacterial population. So bacterial. So I will try to give you some very simplistic, very basic models about self-organization in bacterial population, and it will be done through kinetic models. So it's somehow a continuation of the work by with Nicolaus Bournaveas, Benoît Pertam, who is in Paris, and also a group of biophysicists in Paris at Institut Curie. And so this, all the data I will show in this talk are due to Jonathan Saragosti, who is now currently finishing a PhD. So the outline of the talk is the following. So I will start with very short mathematical preamble about Keller-Segel and very few things that you probably already know for most of you. I will do a very short biological preamble about chemotaxis of Ecole, so how it moves and how it fills some chemical cues. Then I will focus on two modeling issues which are in fact related. So first one, which is about cluster formation, so how bacteria are able to form very stable stationary patterns in some medium. And then I will focus on another thing which is about traveling pulses, which is somehow a variant of the first issue. So on the contrary to Michael Shelley's talk this morning, there will be no fluid dynamics, because it's not it's a very dilute phase, so there is the density of bacteria is very low, but I will focus really on how they interact through chemical signals. So it's really about chemotaxis. So here's a Keller-Segel model. You have seen it in the previous talk. You probably already know it. So it's about a cell density rho, chemoattractant concentration S, which is a signal, which is released by the cell. So in the very simple case, you assume a Poisson equation. And then you have diffusion and drift. So you have two parameters. You have the number of cells, which is denoted by capital M, and you have a chemosensitivity coefficient, which is chi, which measures in some sense the strength of the interaction. So so this model is really devoted to describing motion of cells, motion of particles. So there is no division, no death, as you have in general for tumor growth. Here it's only self-organization by motion of cells. There is a competition between dispersion and aggregation of cells. So dispersion is due to the diffusion part, and aggregation is due to this enhanced chemotaxis phenomenon. So I would like to say that it's a very rich equation from the point of view of mathematical analysis, and so you can do really very interesting and very strong mathematics from this equation. But on the other side, it's a very rigid model from the point of view of pattern formation. And the main goal of this talk is to try to convince you that this model in fact is not the correct one if you want to deal with bacterial interactions. So it's not a good model, and I will try to give you some other model as simple as Keller-Segel, but which might do the job for at least for bacteria. So a brief outline of the behavior you expect with such models. So it depends on the space dimension, as it was mentioned by Nicolaus in the previous talk. So in dimension I of n2, there is a dichotomy, which is very nice in dimension 2. So you have either global in time existence, and more than that, you have decay solutions which are decaying to zero, which are diffusing in some sense. If the initial data is small in some sense, which means that this parameter times time S is small in 2D, for instance. So in this fact, everything happens more or less as if you could simply drop this term out. So only diffusion is acting for small data. On the other hand, if data are large in some sense, then chemotaxis really acts. You have, in some very weak sense, pattern formation, which here means you have blow up in finite time. So it's very crude. The density becomes unbounded after a finite time when the initial data is large. So this is for dimension larger than 2. And this is where all mathematical analysis is really interesting. Try to derive this, to sharpen those constants. It took a lot of time and a lot of efforts. But for this talk, I will focus on the 1D case, which is much simpler, which seems to be less interesting, but still I would like to mention a few things about that. There is no dichotomy in 1D. There is always global existence. It never blows up. There exists stationary state for any parameter, so no dichotomy in this case. And the reason is that the nonlinearity is very weak in 1D, and on the other hand, the confinement due to this chemical drift, this chemical potential is very strong in 1D because the Poisson equation has a modulus of x. The kernel is modulus of x, so it's really strong confinement, and this yields a stationary state, which are explicit. You can compute it, so it's a dilation of this profile with the dilation factor is chi m, so chi times the mass. So in fact, there is a reduced parameters which tells you what is the size somehow of this stationary state. And so it is unique up to translation, and you can prove that you get convergence to this stationary state. For suitable initial data, it means it's globally stable. And you can prove it using the gradient flow structure associated to this problem. So this problem has a very nice gradient flow structure. It's a gradient flow of an energy. And you can prove that the distance, in some sense, I will not comment on that, the distance in some sense towards the equilibrium is strictly decreasing up to the equilibrium itself. Okay, so this tells you that in 1D, things are much simpler. You always have stationary state, and it's stable. But yet, it's not the correct model for Ecole. So what is Ecole? Ecole is, as we have seen this morning, is something like a road. So it has flagellas, up to 5 or 6 flagellas for each bacteria. So it has 2 phases in liquid medium. This is a swimming bacteria in liquid medium. And it alternates, it switches between these 2 phases. So in one phase, the flagellas are coupled in a bundle, and it moves counterclockwise, and then the bacteria swims more or less in straight direction. So this is a swim phase. And then sometimes there is 1 or 2 or more flagellas which decouple from this bundle, which leave the bundle, and then the bacteria will rotate somehow, and it will change direction, and then it will go back to the swim phase. So you have successive swim phase and what is called tumble phase. So this is a run, this is a tumble. And during the tumble phase, which is very short actually, tumble is 0.1 second, and compared to run, which is 10 times longer. So during tumble phase, it will change the direction, and then it will continue in another direction. So I can show you, here is a small movie where you can see from Howard Berg's, when you can see, oops, it goes faster than expected. So if you have a close look, at the beginning you will see here a bacteria with a bundle of flagellas, and you will see 1 flagella which leaves the bundle, and then the cell will go in another direction. So let's do it again. Yes, you can see there is, at some time you have some events which are called tumble, where the bacteria change direction. Okay, so this is a motion of a single bacteria. And then what happens if you put this bacteria in a bath which contains some chemical signals, and in particular gradient of chemical. So heterogeneous concentration of a chemical. And what happens is that bacteria can sense chemical substances. And so it senses chemical species along the trajectories. So it could be either attractant or repellent. Here we consider only attractant, which could be amino acids. that are required for metabolism, or glucose, or oxygen. It could be several things. It doesn't really matter for our purpose. And what is also very interesting for the mathematical point of view is that able to produce those chemicals, which give you a nonlinear feedback, and which give you enhanced chemotaxis as in the Keller-Segel model. And what is known about Ecole is that it reacts not directly to gradient of chemical, because it is in some sense too small to feel to measure properly a gradient. But more than that, it will be sensitive to time variation. So during the run phase, it will be able to measure somehow a difference between the gradient at different points, because it moves, and then it makes some comparison, and then it decides. Either it's better going into this direction, because the time derivative of the chemical concentration is increasing, and then it will go on, continue. It will continue in that direction. On the other hand, if the chemical concentration is decreasing, then it will try to find another direction, another better direction at random. And here this has been measured quite precisely in the 80s experimentally. This phenomenon that E. coli reacts to the time variation of the signal. So chemotaxis really acts on the switch between these two phases. The chemotaxis effect will be sensitive in the switch between these two phases. Okay, so the first, as I promised you, I will discuss two modeling issues, two biological problems. So the first one is about cluster formation. So this has been observed experimentally by the group of Brenner. So it is in this paper in 2003. So they observed stationary pattern of motile bacteria. So I forgot to say, but bacteria are always swimming. They never stop. They are swimming with constant speed, which is more or less 20 micrometers per second. But yet they can produce very stable stationary patterns like that, where you have bacteria swimming around, but globally, when you look for very far away, you see something which is stationary. And with this profile, this is a section from the center of the cluster, which looks like that. So this is the first problem I would like to discuss. And the second one is about traveling pulses, which is quite an old problem in the biological study of E. coli. It amounts to Adler in the 60s, which first observed those traveling pulses. And this is maybe more convincing. This is a picture from Saragosti. So you have channels where you put bacteria initially on the left-hand side. There is some gradient of nutrient like oxygen or glucose, whatever, which goes from the right to the left. So bacteria are likely to travel to the right of the channel. And as you can see here, so this is the density of bacteria. They travel in a very conserved way. In fact, it's a traveling pulse with a constant speed and constant profile. So it's really a remarkable pattern formation issue, which is somehow, if you like, a cluster like this, which is moving to the right of the channel. So here's the traveling. I will show, and this is a dynamic picture of it, and I'm afraid it's a little bit too dark, but maybe you can. Oh, maybe let's try with this one. So this is these little white spots are bacteria, and here you can really see this run and tumble process, where you have one bacteria which is going straight, then it changes, and then it changes again, and you have successive straight runs with reorientation events. And here this is at the level of individuals, single bacteria, and this is at the level of a population of bacteria, where you have the channel which is there. Bacteria are labeled with fluorescent protein, and if you see it well, you could see that there is a band of bacteria which is traveling to the right of the channel. And as you see, all bacteria are performing this run and tumble process. They are always swimming; they never stop. And you could see that denser area is traveling slowly, very slowly to the right of the channel. So this is one of the two problems I would like to discuss. So first, let's start with cluster formation, and for that, let's yes, it's supposed. Actually, it's very easy. It's not very easy, but it has been. It's for experimentalists, they have access to all data, all statistical data for the trajectories, but they don't have access to the chemicals. I mean, they don't really know what are the chemicals involved. But they observe a lot. We have a lot of data about the trajectories, but a very poor data about the chemical. What happens at the chemical level? So I would like to recall the kinetic modeling that Nicolaos has already presented before, although it will be in a different context because this is devoted to bacteria population. So the bacterial density is described as by a density in time, position, and velocity. So you have one more level of description than Keller-Segal. You add the description in velocity to account for reorientation events. So as I said, the velocity space, of course, is bounded, and it's more than that. It's a sphere. It's because they all travel with a constant speed, which is of the order of 20 micrometers per second. And this is a model which has been proposed by Alt and then Hofmer, Dunbar, and Alt in the 80s, which really decompose these two phases. So you have a run phase, which is a transport equation. So without the right-hand side, it would be a free transport equation, which describes cells moving in a straight direction. And then you have a random process in the velocity variables, which accounts for reorientation. So you go from the velocity v prime to the velocity v. So v prime is the anterior velocity, as in the previous talk, and v is the posterior velocity. So this is a Poisson process, which is the intensity is given by lambda, which is simply the integral of this kernel when you exchange velocities, which tells you that you have conservation of mass, conservation of number of cells. And as I told you, the chemotaxis act through the chemical species, the chemical concentration S, and it only acts at the level of tumble. It drives the frequency of reorientation events. Okay. So for the chemoattractant, we hope for the simplest possible equation, which is this reaction diffusion equation. You have diffusion, you have production by the cells. So now rho is the integral of the density with respect to velocity, and you have decay with alpha. And I have to mention that at the level of bacteria, it's quite important to keep the full parabolic equation because the diffusion of the chemical is comparable to the diffusion of cells. So it's difficult to justify a quasi-stationary approximation here. So now you have all the modeling part now is in the tumbling frequency. You have to say what is everything is set. You have to define this tumbling frequency. What is it? So we have chosen to our opinion, the most reasonable one, which is due to Dolac and Schmeiser and Herban and Hofmer, which is to say that this for bacteria, for Ecole at least, this tumbling kernel, which drives reorientation, is a function of the temporal derivative along the trajectory. So V prime is the trajectory of a cells before turning. It measures somehow using, it's known, but from a very phenomenological point of view, it measures the time derivative of the chemical along the trajectory. So this is the material derivative. And it's, you have kind of post-process, and the bacteria interpret this as a tumbling frequency. And the function psi is quite important, and it's a decreasing function, positive, of course, because this is a frequency. And it looks like, so I would say, so you have the argument, which is ds over dt, the material derivative, and it's decreasing. And the transition here, which I will denote it by delta in the cycle. It's quite important. It's expected that it's quite sharp for bacteria. They really can sense. They really make a strong difference between between increasing chemoattractant when it's positive and decreasing chemoattractant when this is negative. So they are really able to make a clear difference between these two. So psi is expected to be, this is psi, the function, is expected to be a sharp, a stiff function. So you have other choices, and I will not comment, which are more relevant, which take into account the inertia, somehow the memory effect of the bacteria, but I will not comment on that. So when this tumbling frequency, when psi is a basal rate, let's say psi naught plus perturbation, a small perturbation where chemotaxis is acting, in this case you can perform a diffusive limit. So this is to say that this amplitude here is very small. So in this case, which I will discuss later on that, but in this case you expect some kind of drift diffusion equation at the limit, in the macroscopic limit, and then you can do that. So it has been discussed already in previous talks. So in this case, it gives you so a conservation equation for the macroscopic density rho and a flux that you can compute formally, which is a diffusion plus a drift. And so I insist on that. This is in the case where the chemotaxis influence is small. In fact, you have a slight influence, which is questionable. But yet, in this case, this is what you obtain. In short, you have a diffusion minus a drift, rho times, let's bold u of S. To simplify, just for the time being, we assume a Poisson equation for the chemical. And this is the explicit flux that you have, which depends on the microscopic variable V, which is the following. And phi, let's recall that psi is psi is equals to psi naught, let's say the mean frequency rate, plus epsilon phi, and phi is assume that phi is even. So you have psi naught here, and you take the antisymmetric part. So this is phi, which is there. So this phi tells you what is the macroscopic flux u. So if you linearize this, you recover, of course, Keller-Segal, because here you see that if you linearize, there is phi prime of 0, which goes away. There is v square, which is positive. Phi prime of 0, which is negative. With a minus, it gives you a positive constant times gradient of S. So a positive constant times gradient of S is exactly Keller-Segal in this case. But I claim that, in fact, it's quite different because you cannot linearize phi. As I claim, this is expected to be quite a stiff function. So the linearized equation is not relevant here. And in fact, it's quite different from Keller-Segal in different points. So first, it's not the gradient of a chemical potential as Keller-Segal. It is bounded, on the contrary to Keller-Segal. So you don't expect a blow-up, finite type blow-up, for instance. And in some sense, it is more nonlinear than Keller-Segal because of this formula here. So back to cluster formation. What is important is cluster formation. So first, you observe stationary patterns of motile bacteria. And second, what a key observation of this paper is that the typical size of the cluster very slightly depends on the number of cells. It essentially does not depend on the number of cells. If you increase the number of bacteria, you will not increase the size of the cluster, which, if you remember, for Keller-Segal was not possible because in 1D, for Keller-Segal in 1D, although the fact it's not bounded in 2D, whatever, but the size of the cluster in 1D is related to chi m over 2, let's say chi of m. So the mass appears in the size of the cluster. So but this is a key observation. And in a very extreme case, where you put delta equals to 0, you choose a step function here. which is a possible choice for psi. In this case, in dimension 1, when you choose the sign function for phi, so a step function for psi, then the drift is not the gradient but the sign of the gradient, only the sign matters. And in this case, you can easily compute the stationary state, and here you find that the size of the cluster only depends on the strength of chemotaxis. So it's one outcome of this very simple model. And if you look at, so this gives you exponential tails for the, so this is the red profile here, the stationary state. This is initial data. This is a time simulation with which ends up at this profile made of 2 exponential tails. And if you compare to this data, it really matches. I mean, so this is exponential tails, and the decay is comparable to what you expect from this very simple analysis. I mean, the size of this exponential is the same as this one if you put numerics in there. So then you can try to see what is the influence of this stiffness, for which you cannot compute explicitly the equilibrium. And here you see that if delta is increasing, then the profile gets smoother, which is expected. You always obtain a profile, a stationary profile, which seems to be always stable, and which is smoother and smoother as this delta here, this stiffness is decreasing, right, when delta is increasing. So a quick observation in for this model, which enables to analyze, to catch the behavior, the long time behavior of this system. In fact, you can reduce this system of 2 equations to a scalar conservation law for the, if you write everything with the gradient of the chemical. So if you introduce eta, which is the gradient of the chemical, you can write a scalar conservation law, which is this one, where eta is non-increasing, because the derivative is minus rho. So eta is non-increasing. F is concave, because you can compute the second derivative, which is a negative thing. So you are in a good shape, and you can prove that there exists always a stationary state to that, and that they are stable. And this is the expression of the flux capital F, which is the integration with capital phi, which is a primitive of phi. Okay, so as I told you, it's questionable to do the diffusive equation, and I will give you some data at the end, which gives some argument in this direction. So you may want to go to the full kinetic model. So first, what you would like to do is the simplest kinetic model, which is 1D in space and with only 2 velocities. So either you go to the right or to the left direction. So this is the full system. So you have bacteria traveling to the right, so with velocity +1, and they switch from velocity +1 to -1, depending on the velocity and the time derivative of the grade of the chemical that they are able to measure. And the same for velocities going to the left. And again, you assume that psi is a basal rate plus phi, which is the antisymmetric part. And you can prove, using the same argument as before, that there exists a stationary state for this this problem. You always have a stationary state, as before. It's more or less the same, actually. And you look for linear stability. So formally, this is how the linear stability problem looks like for J and C, which are perturbations of the stationary state. And here there is some thing which is related to this scalar conservation law trick, that you can reduce this 3 equation sets to only 2 equations, which this is a conservation equation for U, which is not a density, but which is let's say the minus the opposite of the gradient of the chemical, or the perturbation of the chemical, say. And omega is some weight which is related to the stationary state. And this is how it looks like. And for example, when phi is when psi is a step function, when phi is minus the sign, then this weight is twice the sine of x. So this, you are reduced to that. And for that, you can prove that u and j goes to 0 exponentially fast. And this is related to the talk of Christian this morning. But as it is very simple, because you have only two velocities, you can do it directly by computing some useful identity. So if you multiply by the correct rate, which is related to the macroscopic equilibrium, this is what you get. So you have, in the dissipation here, you have one negative term and one positive, one good term and one bad term that you can combine with another relation, which is this one. And this one, which is a good term, it helps you using some Poincaré inequality to control everything and to go. So this is very much related to what Christian showed this morning in a very, very crude and very simple setting. And you can develop the same kind of thing in this case. So you expect at least linear stability of the stationary state, which represent this cluster for bacteria. So for the few minutes which are left, I would like to go back to the traveling pulse problem. So you have a bunch of bacteria which are on the left side of a channel. So what you assume, that you cannot really measure, but you assume it's expected in some sense, that they secrete a chemoattractant, which could be glycine or aspartate or whatever, but you expect something like that. And they also consume a nutrient, which could be oxygen or glucose. And what you observe is that they travel to the right with constant speed and constant profile. That you can see here is slightly asymmetric. There seems to be more cells on the right at the front of the pulse than at the back. So again, you choose a kinetic model. So what you expect is that they will gather due to a secretion of a chemoattractant, as for the cluster formation. But in addition, there is another chemical which comes into the game, and they will consume it, and this will create a gradient, and then this will trigger the motion of the cluster, of the pulse. Now it's a pulse. And you assume the simplest possible kinetics for the chemical species. So S is the nutrient, the chemoattractant, N is the nutrient which is consumed. And now bacteria react to both S and M in an independent way. We cannot do better than that. And I would like to mention that same kind of story has been developed very recently independently by people in Oxford, Edinburgh, Korea, I think, and I think he's in Minneapolis. So again, you can do a diffusive limit. So same story, you end up with the same fluxes, but now you have two fluxes. One which is responsible for the concentration, for the aggregation, and one which is responsible for the motion of the pulse. This is what you get when you go to the numerics of the parabolic equation. So you put this, you know more or less all the parameters except the details of psi. You know more or less the shape, but you don't really know the details, so you have to fit with the data. But when you do this small step, this is what you end up. So these are numeric, so you end up with a traveling wave, a nice propagating wave, nice propagating pulse in the channel. And this is the experimental data, which also should convince you that it's really a very nice traveling pulse that they have observed in experiments. This is when you try to compare the numerics with the data. So the data are the dots at several snapshots. So this matches well, except this, because here the experimental pulse is going a little bit faster, but it's a small correction, small fluctuation in the speed. You have strong dispersion effects when delta increases. As you expect, the size of the cluster will increase. And in fact, in this case, the nutrient effect, the chemotaxis due to nutrient, will be stronger than the attraction, and then it will break the cluster somehow, and then you expect strong dispersion. And in this case, you don't really observe a traveling wave. And this corresponds also to some experiments. And some time, when for example, when the nutrient is limited, when it is all consumed, when the pulse has gone, then you observe a coexistence of a stationary state, which is here is a cluster in some sense, which is stationary, and a traveling pulse. And the pulse detaches from the cluster and it travels on its own. So this is what you expect from this model. You can compute everything in the very extreme case of step function with a sign. You can derive a formula for the speed of the pulse. And what is interesting, so you get these two exponential tails, which were there. Here you have two exponential tails, which are dissymmetric now due to the additional chemoattractant. You can compute an important factor, which is the asymmetry of the profile, which is the ratio between these two exponential decay. And it tells you that the profile is strongly asymmetric when the speed of the pulse is stronger in front of the speed of chemical diffusion. In some sense, this scales like a speed. It's not really clear that it's a speed of chemical diffusion, but it scales like a speed, and it depends only on diffusion. And as I promised you before, this is to convince you that the macroscopic limit is not always relevant. So here is the shape of the front, as before in purple. And here what you see is a bias. So in the blue is more or less the mean psi naught, so the mean tumbling frequency. In fact, it's the inverse of the tumbling frequency, but it doesn't really matter. And in green and red, you have the amplitude. You can see the amplitude as it has been measured of this response function in some sense. And here you see that the amplitude is not that big as compared to the mean rate of tumbling. So this is in some mutant experiment with some mutant, but in the wild type you can see that you have a strong amplitude here for which you should not do the diffusive limit, because now chemotaxis is not a small bias, but it has a big influence. This is between bacteria which go to the right and bacteria which goes to the left, so it has a big influence, as you can see. And in this case, you should go back to the kinetic problem, and here you still observe traveling waves. So this is when you compute numerically the full kinetic problem for the traveling pulse modeling problem. So for different set of parameters, so here you have two sets of parameters. The difference between the two sets lies in this delta, this stiffness, which if it is large, you have a strong dispersion, as we have seen, but still it looks like a traveling wave which is speed down. And if it is small, if the response function is very steep, then you have a strict traveling pulse. And in this case, you can try to compare what you obtain with the experimental data. So dot are experimental data, and as you can see, it matches quite well with the data. So it seems to be a good possible explanation of this mechanism. Okay, and I think I'm done, and I'd like to thank you very much for your attention.
Again, we have time for a very short question.
You said one shouldn't do the diffusion approximation, but maybe one can do hydrodynamic approximation.
Yeah, that's a good. So I try to think about that, but it's not clear in some sense. In some cases, you see here, it's not really clear on that, but you can see it on over. But on the right of the profile, you have really a small bias, and on the left, you have a large one, a large amplitude. So it seems that you could do some hydrodynamic limit on the left, some diffusion limit on the right, and then all the story is to match these two, because this is where the thing is traveling. So everything happens when you try to, but it could be a possibility. I try to do the full hydrodynamic limit, but it's not it's not possible. I think here it's really a kinetic. You really have to keep the kinetic model. But it's a good, very good point.
Okay. Thank you, Vincent.